Hello, College Biology. This is Enzyme Activity Part 2, and we are going to discuss the general steps for enzyme activity. In this diagram, we have our substrate, fructose 1,6-bisphosphate, which happens to be a sugar. We've got this five-pointed carbohydrate. It's in its ring form, and there happens to be some phosphates that have bound to this sugar, which gives us this part of the name, 1,6-bisphosphate. So that's our substrate. Our enzyme is going to be called fructose 1,6-bisphosphate aldolase. That's its name. And the first step is that the substrate is attracted to the enzyme based on compatibility of shape and polarity. So the polar circumstances allow these two to bind. So now we have this situation. Well, what happens once they have bound? Well, the enzyme isomerizes. It changes its shape. You can see that here. So we've got this nice cup shape here and the polarity that matches this. But now that is changing. The polarity of this causes the enzyme to change its shape, which then breaks the bonds in the substrate. So now we are making our product. That is occurring right here. So first we attract the substrate, then the enzyme isomerizes to make the product. Well, the product is then released. That's step 3, and that's what we're seeing down here. We have this shape is this molecule, and then we've got this shape over here, which is this molecule, and so these are our products. So the products are released. That's the third thing, and finally our enzyme renatures back to its original step. So functional enzyme attracts substrate, changes its shape to make the product, releases the product, and then finally renatures back to do it all over again. So in this way, enzymes are recyclable. So those are the steps of enzyme activity. There are, however, conditions and circumstances that can prevent an enzyme from functioning. And we call that enzyme inhibition. The word inhibition means to stop, to restrict, to reduce. So we are preventing something. And in this case, we are preventing the enzyme from working. And there are several ways to inhibit an enzyme. We're going to go over 2 of them. And the first we're going to call competitive inhibition. We've got our enzyme. It's got an active site. It's got a substrate. This is normal. The substrate bonds. Now this model is not showing the enzyme changing shape, but the enzyme isomerizes to create this product. So that's not being illustrated, but we do have the release of the product. This compound has been digested into these 2 different compounds. So normal situation. Down here we have an inhibitor, which is often called a blocker. Now, this blocker has a similar polarity, and one of its regions has a similar shape as the substrate. When it binds to the active site, it shuts the enzyme down because any available substrate in the environment can no longer bind to that active site. The enzyme is not going to isomerize in an effective fashion, and it could get stuck with that blocker in its active site, rendering the whole thing inoperative. So sometimes blockers can, get detached, but sometimes they just stay and ruin the whole enzyme for the rest of its life, and then that enzyme will end up getting digested and its parts will be recycled. But either way, this reduces the effectivity of the enzyme, and whatever reaction, the making of this red Pac-Man and this yellow diamond, that's not going to happen as much because some of the enzymes are being inhibited by the competitive blocker. Now, there is a second type of category. We're going to call it non-competitive inhibition. It kind of works the same way. We've got this substrate binds, enzyme isomerizes, which they kind of leave out, and it makes the product, which should then become released. So that's what we're seeing here. In this case, the blocker does not go to the active site. It does not compete with the substrate for the active site in non-competitive inhibition. Instead, it binds elsewhere, but because of the complex polar dynamics of polypeptides, when you change the polar interactions in one region, it can cause a ripple effect elsewhere in the polypeptide to change regions elsewhere in the protein, and that's exactly what's happening here. When the non-competitive blocker binds to the enzyme, it causes a change in shape for the active site. So even when the substrate can get to the active site, it no longer has a compatible polarity or shape to induce that binding. And so the enzyme cannot hasten that chemical reaction, and the enzyme does not make the product. And so that's our competitive inhibition and our non-competitive inhibition. I hope this has helped. Struggle well.